So this week's topic is going to be on gram staining. Now that we've kind of talked about simple stains, which only involve one dye in order to stain bacteria to show size, shape, and arrangement, we're going to advance into differential staining, which is a bit more complex. So differential staining is the process of using two or more dyes to stain different parts of a cell or different types of cells. And in microbiology, we have three common differential stains. So we have the acid-fast stain, which will show us if the bacterium is acid-fast positive or acid-fast negative. If it's acid-fast positive, it'll stain a bright pink color. If it's acid-fast negative, it'll stain blue. Then there's the endospore stain, which shows us active vegetative cells that stain pink, and then dormant endospores, which will stain green. And then what we'll be talking about today is the gram stain, which shows us if bacteria are gram positive or gram negative. The gram positive bacteria will stain purple, and the gram negative bacteria will stain pink. So differential stains allow you to tell the difference between something, whether again it's two different types of cells or different parts of a cell. So with the gram stain, what it is actually going to do is tell you about the cell wall of a bacterium. Bacterial cells have a cell wall, and sometimes it can be thicker and then sometimes it can be thinner, but the gram stain allows us to identify those cases in which it's thicker or thinner. The main function of the cell wall is to give the cell kind of shape and structural support. It's kind of like the skeletal system, but for a cell. It also allows the cell to kind of take in water and then expand without bursting. Obviously, it can only take in so much water before it will burst, but cell wall gives it a bit more flexibility with the intake of water. So the cell wall is composed of a chemical known as peptidoglycan. Some bacterial cells have more peptidoglycan than others, but they all have peptidoglycan in their cell wall. It's a chemical that is exclusive to bacteria. And because it is exclusive to bacteria, it's often the target of antimicrobial agents because they would be able to target something that only bacteria have. So penicillin works by destroying peptidoglycan in the cell wall of bacteria. That typically causes the bacterial cells to burst through cell lysis because at that point, without the peptidoglycan in their cell wall, they wouldn't be able to maintain their shape and structure anymore. Also, lysozyme is a chemical that we have in our tears, sweat, and saliva. It works the same way. It destroys peptidoglycan in the cell wall of bacteria, causing the bacterial cells to eventually burst through cell lysis. So there are definitely some differences between gram-negative and gram-positive cells, and it is important that you understand these differences. So gram-positive bacterial cells tend to have a thicker cell wall, which basically means they have a thicker peptidoglycan layer, since that's what the cell wall is made up of. It's not important that you know the exact thickness, but they do have a thicker cell wall. Gram-negative bacteria typically have a thinner cell wall, meaning that they have less peptidoglycan. We'll see in a second, but gram-positive bacteria, when you gram stain them, they will stain purple. Gram-negative bacteria, when you gram stain them, they will stain pink. Gram-positive bacteria contain chemicals called teichoic and lipoteichoic acid in their cell wall. It's actually embedded within the peptidoglycan and helps maintain that peptidoglycan. And then gram-positive bacteria also, they do not have an outer cell membrane. Bacteria all have a cell envelope that's made up of multiple layers. Gram-positive bacteria do not have the outer cell membrane that is sometimes found, or that is found in gram-negative cells, so they lack the outer cell membrane. Whereas gram-negative bacteria, they do have that outer cell membrane in their cell envelope, and we're going to see images of that just in a second. Also, gram-negative bacterial cells have these lipoproteins and lipopolysaccharides. These are just fatty proteins and fatty sugars that are found in the outer cell membrane. We'll talk about them much later on the semester, but they are often can serve as toxins in the human body and cause things like septic shock and diarrhea and vomiting, all different types of stuff. So this is the cell envelope that I was talking about. So all bacteria have a cell envelope, and it's basically this outer layer that surrounds the bacterial cell, and it's made up of several layers in itself. So gram-positive bacteria, they only have 2 layers in their cell envelope. They have the inner cell membrane, which sometimes gets called the plasma membrane, and then they have their cell wall, which is composed of a thick layer of peptidoglycan. So that's it. They just have those 2 layers in their cell envelope. Gram-negative bacteria, though, they have 3 layers in their cell envelope. They have the inner cell membrane, they have the cell wall with a thinner layer of peptidoglycan, and then they have the outer cell membrane. So they're different in the sense that they have 3 layers in their cell envelope, whereas gram-positive bacteria only have 2 layers in their cell envelope. And we can see this here again. This whole structure is the cell envelope for gram-positive bacteria, gram-negative bacteria. So the cell envelope for gram-positive bacteria is going to have the inner cell membrane and then the cell wall with a thick layer of peptidoglycan. And we can even see those teichoic acid and lipoteichoic acid chemicals in there that are help maintaining all that extra peptidoglycan. And then with the gram-negative bacteria, this is their cell envelope. It's made up of 3 layers: the inner cell membrane, the cell wall with a thin layer of peptidoglycan, and then that additional outer cell membrane. And we can even see those lipoproteins and lipopolysaccharides embedded in that outer cell membrane that could potentially serve as toxins. So the main point of a gram stain is basically to decipher the characteristics of the cell wall of the bacteria. Does it have a thicker cell wall with more peptidoglycan, or a thinner cell wall with less peptidoglycan? And when you gram stain, you can tell the difference based on color. If it turns pink in the end, we know it's gram-negative. It has a thinner cell wall with less peptidoglycan. If it stains purple in the end, we know it's gram-positive. It has a thicker cell wall with more peptidoglycan. So when you do the gram stain, obviously the first thing that you would go through would be the whole smear prep process as far as prepping your slide, which you learned last week, with the small drop of water and then adding the bacteria and then letting it air dry and heat fixing it and then letting it cool down. And at that point you would start with the staining procedure. So the first step with a gram stain is you would add crystal violet. That's the first stain, the primary stain, and you would let it sit on there for 1 minute. And at that point, everything would stain purple. So whether the cells were gram-positive or gram-negative, initially they all stain purple. Then you would rinse the excess dye off with water. The second reagent you use is Gram's iodine. Gram's iodine, although it's orange in color, it doesn't actually stain the bacteria. All it does is act as a mordant or a sealant. So it kind of seals that crystal violet color in. So you would add that for 1 minute and then rinse with water. And at that point, everything is still purple, it's just sealed in. The third step, the alcohol step, is the most important step. This is the decolorization step. This is the step that separates the gram-positives from the gram-negatives. So when you add alcohol to the slide, the gram-positive bacteria, because they have a thicker cell wall, they are able to hold on to the purple color, to the crystal violet color, even when you rinse the slide with alcohol. So they are actually going to stay purple. The gram-negative bacteria, though, because they have a thinner cell wall with less peptidoglycan, they're not able to form a tight bond with that crystal violet color. And when you rinse the slide with alcohol, the gram-negative bacteria are actually going to get stripped of all their color, and they are going to be clear for the time being. So at this point, your gram-positive bacteria are purple. Gram-negative bacteria are actually clear because they weren't able to hold on to the purple due to their thin cell wall. Then we would rinse with water, and our very last step is to add a counter stain known as safranin, which is a pink dye. The safranin is going to basically stain anything that had lost its color in the previous step. So basically, it's just going to stain those gram-negative bacteria pink. So you would just add that for 1 minute and then rinse with water. And then in the end, if your bacteria were gram-negative, they will be pink. If they were gram-positive, they will be purple. And I know some of this is hard to visualize, so there is a video in D2L that you can watch that actually goes through this entire process. Like I said, that decolorization step is so important, and it is the step that is frequently messed up. But that is the step that's going to separate the gram-negatives from the gram-positives based on the thickness of the cell wall. Because remember, if the bacteria have a thicker cell wall, they're able to hold on to the crystal violet when you rinse them with alcohol, and they'll stay purple. But if they have a thinner cell wall, they're not able to hold on to the crystal violet, and they'll get stripped of their color when you rinse with alcohol. So what's complicated is adding the appropriate amount of alcohol, because if you add too much alcohol, if you over decolorize, you can actually strip the gram-positive bacteria that were supposed to stay purple of their color. And if you strip them of their color, the very last step is to counterstain with safranin. Your gram-positive bacteria could potentially stain pink and look gram-negative if you use too much alcohol. But then it's also confusing because if you don't add enough, you could under-decolorize, meaning those gram-negative bacteria won't lose all their purple if you don't add enough alcohol. And in the end, they might still look purple when they were supposed to be pink. So you do have to add just the appropriate amount of alcohol. It doesn't have a time limit like our other stains with 60 seconds. The crystal violet, the saffron, and the iodine, those were all 60 seconds. With alcohol, we typically just add 1 drop at a time until the runoff is clear. And once the runoff is clear, then we immediately rinse with water. So it's going to be different for every single slide, but it's important that you don't add too much or too little alcohol. Also, anytime you do a gram stain, you want a fresh culture. We typically don't want to work with bacterial cultures that are more than 24 hours old. The reason for that is with gram-positive bacteria, after 24 hours, their cell walls start to break down. And when their cell walls start to break down, that means they're not going to be able to hold on to that crystal violet color like they should. So if you're working with a really old culture and you're doing a gram stain on it, and it's supposed to be gram-positive, but it keeps coming up pink, that's probably because the gram-positive bacterial cell walls have already broken down. So it's really important that you always add an appropriate amount of alcohol, not too much, not too little, but also that you're working with fresh cultures. So what else could possibly go wrong? Well, if you follow the procedure, nothing, but there are some few mistakes that commonly do happen. Should you forget to use iodine, remember iodine is the sealant. It seals the purple color in. If you forget to use iodine and then you rinse with alcohol, that crystal violet is not going to be sealed into those gram-positive bacteria, and the alcohol will strip the color from the cells. So they would look pink in the end, just like the gram-negatives. So it is important that you add that iodine for 60 seconds. Let's say you completely forget to do the alcohol step. You just do crystal violet, iodine, and then straight to safranin. Remember, the alcohol is what pulls the purple out of the gram-negatives. If you don't use the alcohol, the gram-negatives are going to look purple just like the gram-positives, so everything would be purple in the end. If you forget to apply safranin, which is the very last step, so you do crystal violet, you do iodine, you do alcohol, but then completely forget about safranin, well, your gram-positive bacteria are not going to be affected. They're still going to be purple. But those gram-negative bacteria aren't going to have any color. Remember, they lost all their color when we rinsed them with alcohol, so they're clear. The last step is to stain them with safranin. If you forget to use safranin, you're not going to be able to see them under the microscope. They're actually going to be colorless. Reversal of crystal violet and safranin. You kind of automatically think, oh, if we just reverse the order of the stains and do safranin, iodine, alcohol, crystal violet, it's just going to reverse the order of our results. So gram-negatives would be purple, gram-positives would be pink. That's not actually what happens, though. Iodine only works with crystal violet. So if you did safranin first and then iodine, and then what would happen is the iodine wouldn't seal in the safranin because it only works with crystal violet. And then after iodine, you did the alcohol, all the color would be stripped of all the cells and they would be clear. And since your last step is now to stain with crystal violet, all cells would be purple in the end. So it doesn't work exactly how you might have thought it would work.